Western blotting is a fundamental biochemical technique that we can use to see if there are specific proteins in a sample. And it builds off of a technique we talked about before, SDS-PAGE, in which we used electricity to get proteins to move through a gel mesh and separate by size. So with SDS-PAGE, you can separate the proteins by size, and then you have these proteins locked in a gel. So the question is, what do you want to do next? You can do something like a Kamasi stain, which will stain for all proteins, and you'll get a bunch of bands. But you won't know what those bands are. So Western blotting allows you to see what those bands are. Well, kind of. You have to know what you're looking for. You basically go fishing for specific fish using antibodies. So I'm going to give you an overview, and then we're going to go in more detail on each of the steps. So basically, the idea is that you take proteins from different conditions. So maybe you have cells before you add a drug and cells after you add a drug. Then you're going to start off like we did with the SDS-PAGE. You're going to, so you need to break open those cells, isolate the proteins. Then these proteins are all going to have different charges and sizes. And so basically you're just going to use the detergent SDS to unfold all the proteins, keep them soluble, and give them a negative charge that's going to allow the electricity to get them to travel through the gel mesh. So that's the gel electrophoresis step. But then you have your proteins locked in this gel, which isn't very useful for Western blotting because you're going to have to be doing a lot of binding and washing and binding and washing. And these gels are really thin and they're just going to rip. So what you need to do is you need to transfer them to a more sturdy membrane. So this is the blotting step, or the transfer step. And there are different ways to do it. I'm going to tell you about the way that I've done, which is wet electrotransfer. So at this point, you have your proteins in the gel, and you need to be able to see if there are specific proteins in there. But so for reasons I will talk about, you first need to block the membrane. So basically right now, your proteins are on this membrane that is basically kind of like duct tape for proteins. So it's going to stick to basically any protein. And so we want it to stick to the proteins that we've just transferred, but they're already there. But the protein, we're going to use antibodies, which are proteins, as probes to see if our protein of interest is there. So the antibodies, if there are free spots on that sticky tape membrane, they're just going to stick, and then you're going to get high background. So we don't want them to stick everywhere. We only want them to stick where our proteins are present. So we block the membrane using a generic protein and some detergent to kind of make things more slippery, so that the antibodies can't just generically bind. And then we're going to bind with a primary antibody that's going to recognize directly the thing you're interested in, and then a secondary antibody that's going to bind to that primary antibody. But it's also, the reason for this is that this way you amplify your signal, because you can have a bunch of secondary antibody binding to the primary antibody. And the secondary antibody is attached, or what we call conjugated, to something that's going to make it able to visualize it, such as some fluorescent protein. And then you're going to wash. Yeah, there are a lot of washes because you want to make sure that you're only getting the specific things, washing away anything non-specific, because you flood the membrane with antibodies, and so you want to get rid of those antibodies before you go to screen it, and then you visualize it to see if your protein is there and the relative amounts. So that's the overview we're going to be returning to throughout, and I'm going to show you the slides, the steps in more detail. So starting with this beginning part, so we're going to take our cells, and you don't have to start with cells. If you can start with like some purified protein mixture or that sort of thing, but often you're starting with cells, so you want to lyse them, which is where you break them open. And so now you get this mix, and you get rid of the membrane parts by centrifuging. So you spin them really fast so that the membrane parts pellet out, and now you're left with this mix of soluble proteins. But they're all folded up, and they all, if you try to send them traveling through a gel, because they all have these different shapes, they're not going to uniformly travel based on their length. So it'd be hard to compare their sizes. Also, the way that we get the proteins to move through the gel, so the electrophoresis part, it relies on the proteins being negatively charged, because basically you use a charge gradient where you have a negative charge at top that's going to repel the negatively charged proteins, and a positive charge at the bottom that's going to attract the proteins. So we want all the proteins to be negatively charged and unfolded. So the detergent SDS is really great for this because it has this long, like greasy, non-polar tail, and this hydrophilic, water-loving, negatively charged head. So when the protein unfolds, it kind of like globs onto the protein and keeps it soluble in its unfolded state, and also gives it this negative charge. So this is going to allow us to separate the proteins by length in this gel electrophoresis step. So basically, in this first step in SDS-PAGE, we're sending the proteins traveling vertically through this gel. And you can see in this picture that they're separating by size. So basically what happens is the proteins are kind of going to get tangled up in this gel mesh. And the longer they are, the more tangled they're going to get. And so the slower they're going to travel. And so when you turn off the electricity and prevent them from moving through the gel, the bigger ones are going to be higher up. So what's happening, that's what's happening. But what you're seeing is basically nothing. You're just seeing the tracking die. When you're doing a Western blot, you probably have a stained ladder though, that you'll be able to see, and that stained ladder is going to be important for making sure that your protein's transferred to the membrane. So you start the same way, whether you're just doing like a normal SDS-PAGE or you're doing a Western blot. So with a quote-unquote normal SDS-PAGE, what you might do is Kamasi staining, which is an all-protein stain, and so you're going to see bands for all the proteins that are present. But you don't know what those bands are. So you can get a general idea of their size, especially if you use a protein ladder, which is basically a mix of proteins of known size. So you can see that on the left-hand line. So that allows you to compare the protein sizes, but it doesn't tell you what they actually are. So for that, you can look for Western blotting, which is what we're going to talk about now. And that's going to show you specific proteins, and it's often with like some fluorescent readout or whatever. So you might get something that looks like this, and you can see the thicker bands correspond to more protein, but it's not like very quantitative or anything. Also, an important thing that I'm not showing here is that you're going to want to like also stain or also probe for a protein that should be the same in all the samples, like some housekeeping protein like GAPDH or tubulin or something, that's like a loading control, so that if you have a stronger band, it's not just because you loaded more. If it was just because you loaded more of that sample, then you would have more of that loading control as well. But if you have the same amount of loading control but more of your protein of interest, that means that the protein is like being expressed more or degraded less or something in those cells. So first we need to transfer it. So we're going to blot the proteins. We're going to move them to a membrane. So in SDS-PAGE, we had the proteins go down, and now we're going to have the proteins come out. And so this is showing a wet electrotransfer setup. And so basically, these use the same electrophoresis module, like the bucket outer part, but a different like insert module part. But there are different ways of electrotransfer. There's like dry and semi-dry and stuff. So and some of those are like horizontal and stuff, so I'm not going to talk about those. So this is the way that we do it. And basically what you're going to do is you have this sandwich you make this like sandwich with gel and membrane and sponges and filter paper, and you're going to stick it in that module, and it'll so this way, instead of, it's confusing because it looks just like kind of like the proteins would be going down, but the way that the electrodes are set up, the charge gradient is actually going like horizontally. So you're taking things out of the gel onto the membrane. But in order to do that, you need to make sure that you set up your sandwich right. So basically, you have this like cassette, and you want to make sure, so you have filter papers to help keep things wet and wick the buffer through, because the important thing is that you need to have the charge be able to flow consistently. Like you need to have you don't want to have any bubbles or anything, or any dryness where the electricity can't travel through, or you're going to get weird artifacts on your gel and things aren't going to transfer. You need to have like a conductive path for these proteins in the charge to move. So that's why you have the filter paper, and why it's important that when you're making your sandwich, you want to make sure that everything stays wet the whole time, so you like make it underwater and stuff. So after your filter paper, you're going to have your gel, which has your proteins for now, and then you have the membrane you want to transfer them to, some more filter paper, some sponges to keep things snug. You don't want things shifting around, or else the proteins aren't going to be where you expect them, and you don't want things just like diffusing out. But the most important thing is that you put your cassette into the thing in the right direction. So you want to make sure that you have the membrane in front of the gel in the way that the electricity moves in your module. So once you set it up, you're going to wrap it all up and slide it in. Fill up the tank with your transfer buffer, hook up the power. Then you're going to get something where when you open this sandwich thingy up, your gel should be empty, and the protein should be on the membrane. You're not going to see the proteins, and you're not going to see them in the gel, and you're not going to see them on the membrane. But what you can see is the pre-stained ladder. So normally when we're just running like a normal SDS-PAGE, we'll use a cheaper ladder, which isn't pre-stained, and you just stain it the same as you do your proteins, and so you're just relying on the loading buffer to track the run. But here it's important that you see that the proteins actually get transferred. So if you use a pre-stained ladder, then that pre-stained ladder is going to get transferred from the gel to the membrane along with all the other proteins. So you shouldn't see the pre-stained ladder should all be on that membrane and not on your gel. The amount of time that you want to use to transfer is going to depend on like how big your protein is. The bigger it is, the longer it's going to take to travel, and you might need to like up the voltage or up the time or something. But if you do that too much for a smaller protein, might actually like come out of the membrane, so you have to kind of optimize for your protein of interest. Then to find that protein of interest, you're going to use antibodies. So now we're at this stage. So an antibody is basically like a little protein that recognizes a specific part of something else. And so we call that something else an antigen, and we call the part that gets bound by that antibody an epitope. And so we say like recognize, but really that just means that they bind specifically to it, or you hope it's specific. Sometimes they can be non-specific, and then you can get noisy signals. So I like to think of it kind of like Where's Waldo. So each of these antibodies has like a constant region, which is in this case they're different. So there are different types of antibodies, but and we're going to talk about these IgG antibodies where they have this like Y shape. And so the base of the Y is like a generic part that's specific for that species. And then the tips of the Ys are the variable parts, and that's where the antigen is going to bind. So those variable parts can recognize different parts of a protein, like recognizing different parts of Waldo. So if you have what's called a polyclonal antibody mix, that's going to recognize, like, you're going to have a mix of antibodies that can recognize different parts of Waldo. If you have monoclonal antibodies, that means you just have one of these antibodies. So this can have better specificity, but lower sensitivity. We'll get to that more later, but basically, if you recognize more parts of Waldo, you're more likely to also recognize similar parts in other proteins, and thus get lower specificity. So you're recognizing other things, and you're getting like false alarm signals. But if you have a bunch of different antibodies, like that recognize different parts, then there are more places the antibody can bind on your protein, and thus you can have a stronger signal. And polyclonal antibodies are also often cheaper. So if you were looking before, we saw there was this like primary antibody and secondary antibody. So your primary antibody is going to have a generic base that's generic for the animal that made the primary antibody. So this could be like goat what's a protein that you're wanting to look for? Goat, we're just going to make up a protein, anti-fabulous. And so we're going to have this protein fabulous. And so our primary antibody is going to be goat anti-fabulous. So the base part is going to be generic for goat. And then at the top, we're going to have that specific part that specifically binds to fabulous protein. So you get something like this. So it's useful because it bound to our fabulous protein, but it's not useful because there's only one per protein and because we can't see it. So the whole point of the probing is to make it so that we can see it. So for this, we need a secondary antibody. So this is going to be made in a different animal. So maybe this is like rabbit. So this would be rabbit anti-goat. So it's going to recognize the generic part of our first antibody. And this is good because we can use it with goat antibodies for any protein. So we can save money because we can use like the same secondary antibody for any antibody that was made in a goat. We just can't use a goat secondary antibody. You have to use like a different species. So we have this secondary antibody, which is going to be recognize the generic part of our first antibodies. And it's going to be attached, or what we call conjugated, to something detectable. So this could be like a fluorophore, so something that's going to give off light when you shine a light of a different wavelength of it, or it can be some like chemical reactant group that if you add chemicals, it's going to undergo some sort of reaction that's going to make it a colored product. So an example of this is like horseradish peroxidase. So the secondary antibody is going to bind to your primary antibody, and then when you add those chemicals or you shine that light, you're going to get some sort of signal that you can see, so often fluorescence or some sort of color change. Sorry, I just backed up. Okay. So that's the idea. But we skipped a step in our explanation, because if you were just to do that, you would get really high background. So you'd have a lot of crying wolf, basically. So as we talked about in the very beginning, our membrane is kind of like sticky glue for protein, or like duct tape for protein. And so our antibodies are proteins. So our antibodies are just going to stick to that membrane unless we block it. And so basically blocking just means we take like a generic protein like BSA, bovine serum albumin, or even milk. A lot of times you make a milk solution with powdered milk, and often detergent. So the buffer is often like TBST, so tris buffered saline with tween. I think it's tween, not trine. But basically that's a detergent. So you can think of it kind of like when your hands are slippery, it's harder to grab things, so it's harder to stick to things. Actually, my cousins and my sister and I, we got locked in a bathroom once because we weren't very smart. We were little at the time, but our hands were all wet and soapy, and we couldn't open the door because our hands were so slippery. So it's kind of like that. Like if the membrane's really slippery, the antibodies aren't just going to bind randomly. Or that's the hope, at least. And then you also have all those wash steps to see, so that if they do bind, then they'll get washed away. And so the wash steps are like the worst part of all of this, because it's just really time consuming and boring, because you keep having to wash and wash and wash. But it's important to wash, or else you get high background. So that's the basic idea of that. Some other things to look out for. So cross-reactivity. So this is what we're talking about with the monoclonal versus polyclonal antibodies. So if you have polyclonal antibodies, there's more of a chance. So that's where you have antibodies that recognize like different parts of Waldo. So it's more likely that you're going to recognize other things that also have those parts, or something similar. So you're going to have higher cross-reactivity. But then you also have the sensitivity, where if you have a polyclonal, there are more places to bind, so you can get a stronger response and signal. But with monoclonal antibodies, there's only one place to find, so you're going to have lower sensitivity. Although with the secondary antibodies, you'll get somewhat of a boost with either method. A little troubleshooting. So yeah, so you want your antibody to only bind the protein of interest. If your antibody doesn't bind, doesn't bind to your protein. This can actually happen. Not all antibodies are made alike. Some antibodies are a lot better than other antibodies. You want to make sure that if you have like a positive control, so a protein that you know is that protein that it should be able to recognize, that's a good, you want to include that. So like if we had to sample a pure fabulous protein, we'd want to include that to make sure that the antibody could recognize it. So antibodies recognize proteins based on their shape, kind of. So in a Western blot, our protein is unfolded. So normally what you want is you need to make sure that the antibody that you're using is can recognize the unfolded form, which usually it can. But like sometimes people use antibodies like in cells, so where the protein's folded, but you need it to be able to recognize the unfolded form. Antibodies can also expire, so you want to make sure that you store them properly, because they're like they're proteins, so they can degrade and that sort of thing. So you often store them at -20 or -80. You can also store them like diluted for shorter periods in the fridge, because you can actually reuse the solution a couple times. So if you're getting high background, you want to make sure that you're adequately blocking your membrane. If you're getting kind of grainy signal, you want to make sure that if you're using powdered milk, you want to make sure that milk is really dissolved, or else you'll get grainy artifacts. And don't use milk when you're detecting phosphoproteins or a biotin-based detection system. So basically, if your milk has a bunch of phosphorylated proteins in it, and so it's going to cause problems if you're trying to stain for phosphorylated proteins. And yeah, so yeah, I think that's basically everything that I wanted to cover today. Sorry that went so long. I didn't want to stop because I figured that people might want this information, and if I tried to do it in 2 videos, then I'd have to re-explain a bunch of stuff and it wouldn't work too well. But I hope that was helpful, and I hope you don't have to do too many Western blots because I really hate them. But some people actually like them, so to each their own, and that's one of the great things about science is that everybody can find their niche. So yeah, I hope that helped, and happy blotting.